So we'll start with Dr. Tirumalesh. There's a small change in the orders just to accommodate a few people who are having overlapping sessions. So he's going to speak on mitochondria, new biological findings in treating retinal diseases. There's going to be a mix of many specialities overlapping here, the dry eye, ocular surface, wound healing. So the whole thing has to be interactive. There are also a lot of people from the industry, people representing companies here. So if you have anything which is linking to your area of interest, you can just, there are mics there, or you can come forward and just ask a question. So there are scientists here who will probably help you to give some answers to this. So we'll start off with Dr. Tirumalesh. Ready?
Yes, sir.
Yes.
Thank you very much, sir, for this opportunity. And my topic is new biological findings in treating retinal diseases. Although the findings are not new, only thing is the way we are looking at the disease is a little new. This is the team that Dr. Rohit had put together and given us a task of looking at DR and AMD in new light. And when clinicians look at DR AMD, this is what we see. We know that there are drusen's and there are underlying RP changes that are there, and they are the reason why the disease kind of progresses when the RP gets stressed with age and with the inflammation. So there is cellular level of inflammation which is going on, and that's the reason. Now, what proof do I have to show is this particular slide. Now, you can see on the right side where those small orange things that are moving, these are activated NK cells, and you see that the NK cells are just moving over the RPE without producing any problem. Now, towards the right of your screen, you see that the RP cells, which got a little stressed, the contact with the NK cells induces what is called as pyroptosis. So in pyroptosis, what the same NK cell which was moving over the RPE without producing any problem kind of eats it up, right? So we are continuously collaborating with our collaborators outside to kind of look into newer treatment possibilities for dry AMD. And this particular collaboration had yielded a few interesting findings. Dr. Stina was working with Professor Ziegler, and they were working on crystallins. As we all know, crystallins are proteins that are present in the lens. But the same crystallins, when they are altered, with age, the protein expression gets wrong and the RPE kind of produces dry AMD, and this is one of the pathways which is now being pursued. This is popularly the mTOR pathway, in other words, and the gene responsible for the mTOR pathway downregulation is crystallin. Now, we all know that AMD does not have an animal model. That is because the genes that have been associated with AMD, if you downregulate them, it either kind of looks into the vitamin D pathway or how lipid is metabolized, because they thought that drusen has lipid in it. But when we kind of knocked out the CRYBA1 gene, we were able to successfully produce AMD in the mice, and you can see that as the knockout continued over 4, 6, and 12 months, we were absolutely able to produce the drusen's. So this is the initial proof that if you downregulate beta-crystallin, the disease can produce an active working model of AMD. So we are pursuing this particular path of alteration of beta-crystallin. Now, what we did was the next step where we were pretty much sure that when you alter the crystallin gene, it is going to work in case of dry AMD. So we took it forward and we did the first non-human primate study in the rhesus monkey. So first thing that you have to prove with any therapy is that it is not going to produce harm. So for that, we injected the CRYBA1 gene and also the AAV vector that we have produced within India indigenously, and in our collaboration, we are using the gene sequencing from the US. And what happened was we were able to show that there is absolutely no inflammation in the monkeys after the injection, and you can see both the OCT and the fundus pictures. We also did a fluorescein. You can see that they are as clear as, after the injection, it did not produce any kind of vasculitis kind of a reaction. So this is sequential imaging for up to 6 weeks. And once the imaging was done, we were supposed to produce histological proof. So we did the sacrificing of the animals, and we wanted to see what we have put within the eye, does it get integrated or not? You can see the green line on the bottom where I put an arrow mark already. So we were definitely sure that the RP picked up this particular thing, and it also was able to produce the protein that we put inside. Now the second part is we did not use the subretinal route because subretinal surgery is something which is very, very complicated and only a few can do. What we did here is we injected in the suprachoroidal space. So suprachoroidal space is kind of an office procedure. It's just like an intravitreal injection. And with that, we were able to produce gene within the eye and produce the protein expression that we wanted. Now we further studied if we put the particular thing in a live cell, does it work or not? Now this is a live cell image. You can see that the RPE cells, we put a transfection, we put the vector and delivered a particular gene fluorescent protein. Now, how it integrates with our particular gene, you can see that it initially starts in the nucleus, the GFP is there, and once the protein comes into the cytoplasm, you can also see that the particular protein is produced. So the vector works, the method works. If we are probably right, then very soon AMD, dry AMD can have treatment which can be used for the betterment of these patients. Now, the other disease that we are closely looking at is diabetic retinopathy. This is a single slide where it has summarized how diabetic retinopathy initially starts as metabolic dysregulation, then goes into subclinical or cellular, intracellular inflammation with those factors that are there, and then it goes into proliferation and tractional detachments as it progresses. Now, the emerging evidence is even in DR, the cells are aging, but they are aging at a faster rate. So when they are trying to save themselves, they are going to produce the VEGF. So it is initially a chronic inflammation within the cell at the cellular level. Then it is going to produce factors which will try and get it vascularization because hypoxia is the predominant change that is happening in the retina. Now every single component of the retina, starting from retinal glial cells, the neuronal cells, the Muller cells, the RPE, actually contributes in producing these small factors like MCP, IL-6, VEGF, and ICAM. Now, am I just talking like that, or is there real evidence in patients? So this is an aqueous humor analysis. You can see that all those factors are present, and at different times, when the patient have different stages of disease or DME, you do have these factors which are kind of varying. Now, DR has the longest surviving animal model. This is from an SD rat. You can see that if you put streptozotocin in its peritoneum, it is going to produce DR. And we were investigating one of the targets, which was ICAM1. So the ICAM1 antagonist that we used was LFA, and it did produce suppression, which was as good as the VEGF. So the safety part is what we are kind of looking into, and the dose escalation part has to be looked into. And you can see that the model absolutely mimics how it is seen in the human patients when it comes to DR. Now, along with ICAM, since we know that the inflammatory factors are going up, now there is a cyclic GMP-AMP synthase which is kind of gets increased, and this will stimulate the interferon genomics. So how does the inflammation start within the cell? Is by the upregulation of the IF and interferon genes. So is there a way that if we kind of alter this, can the senescence be reduced? So that was the paper, and this paper got published, and we showed that the upregulation of the sequestering pathway is the one which produces early senescence. So considering this, altering or conditionally knocking out the sequestering pathway can also be one of the treatment modalities that we can probably see in the future, which can probably alter the way we treat diabetes at different stages of the disease and not just rely on VEGF or giving intravitreal steroids. Thank you very much.
Thank you, Tirumlesh. Swami can get ready here, and like to ask Dr. Dhanashree ma'am to give your comments on this, and Professor Sundareshan also after her. So the question I want to put it to this clinician scientist here is, so say 5 years from today, we're sitting in this kind of a panel. Do you see a more organized form of anti-VEGF? The word VEGF is because first targets were only VEGF, so that's why I use VEGF. So it's more like now we'll have to look at the different forms like LFA1 the antagonist or the ones which can block it are not VEGF blocking ones. So do you see that we will have to really look at the different forms of these customized therapies?
Yes, sir. So what we are trying to look at...
just be very brief so that...
We are just looking at alternatives apart from VEGF. So the ICAM that I mentioned is chronologically that is the first one to start. That is the one which switches on the angiogenic cycle.
Ma'am?
Very good work and wonderful talk. So there is always a focus more towards like looking at inflammatory, basis for all the diseases that we know. So even all these vascular diseases that we have been treating with anti-VEGF, there is some amount of inflammatory, modulation going on. So it is definitely essential that we look at all these molecules and look at the newer ways to and therapeutic targets that we have to identify.
So I think not only this disease, I think I was looking at the previous session by Professor Honnavar and Fairoz here, that every new disease has to be relooked at it. And you also worked on retinoblastoma, and I was just looking at that paper, the Cell paper on retinoblastoma with a new way of how you look at therapeutics. Everything has to be looked at in a different, different light. The time has come to move forward, and that's why when you say anti-VEGF, that means you're working only on VEGF and nothing else. So that is the mind block we have, and everything revolves around it. Different form of isoform, different part of biosimilars. So I think one thing is that we should come out of those name and then you suddenly start looking at it in a different way.
Look at the whole holistic picture of the disease.
Professor Sangban sir, please. So we will move on to a very interesting concept where we will have our team. The whole idea of this session was conceived by Dr. Lalit Verma sir. What he said was we should try to bring in more lively lab-based discussions, and he wanted us to try to link the live lab to the conference halls. So now what we are trying to do is, there are a lot of talks here, many of which from different institutes. Most of them have the same kind of a laboratory experiences. So we thought we'll do a live relay from the lab and see and give a clinician a kind of a peek preview into what happens in the lab and how these scientists are trying to work with the clinicians in different institutes to give back the answers what we are looking for. This includes from the tear film to the retina and the tumors. So if you are ready, then you can link the lab and you keep talking about what each one of these things are happening and how clinicians can keep asking these questions, which will help scientists like you to create models to bring these experiments. And a lot of this is going to be discussed by the group here. They're going to speak about it. Are we ready?
Thank you, sir. Yes. Pani, are we ready? Live? Yeah? Okay.
And whatever you see here is the live video from the lab. So not this.
He's going to have a split screen.
Dr.
Okay.
The Swami heads one of the divisions of clinical research, who he's looking after the whole process of translational immunology and molecular signaling.
Okay.
And let's start.
Thank you. So
we should finish on time.
Yes. So most of the work transferring from the patients to the lab, most of them are based on tear biomarkers. Okay. So how does it look when we collect the tears after you collect the Shermers, when it goes back to the lab, and when we plot graphs like these? Where do they come from is what we wanted to show. And there are two ways it can happen. One is through flow cytometry. So we're going to patch in live from flow cytometry. It is a device which can measure close to 50 different cytokines from a single drop of tear. And Archana will be there on the other side. And of course, what we have also done is to make sure it is easy for the clinicians to have a kit. So we'll have live from both these units, okay? And we'll have one thing on the side to give you an idea of what that is. Flow cytometry is a cell's, facial recognition technology, you can say. So what you see there is a flow cytometer, and she is currently using a tear sample which is being analyzed using a process called bead-based multiplex ELISA. So what happens, this is a 2-day process, at least one whole day goes in this and another day of analysis. And this is the device, and what you see to the right side is the acquisition. Every cytokine gets measured like that before it forms like a dot or a bar in your graph. So, and we can also show that this is a laboratory intensive stage. We can patch in with the biomarker kit. where you can get the very same technology into your practice, and we can patch in with the biomarker kit now. How does it look? So right now you can see a Shermas which is getting shaked there with a buffer which goes into the kit. And what you see right side is a kit already loaded with a previous sample.
And then this kit can load anything. It can load, it's from your tears to CSF, blood, aqueous, vitreous, tumors.
Yeah.
So virtually, it's a pool, it's a well which can house anything in it.
Yeah. And no technical assistance required. You just add, and that's it. You get reliable, quantifiable data. This is both for scientific use as well as clinical use.
So this is something which is, there are, I discussed with a few of my friends from Heidelberg and AIOS that, this is what helps us to look at the tears, and we were talking about how we can correlate this with your healing, basically epithelium or creating scarring or any of these factors. Of course, these cytokines here are limited, but I'm sure he's going to show us the images of the mass spectrometer. People asked, how do you, there are two things. One is you look at the cytokines, and cytokines are, you can have any number of cytokines, but you will have to figure out these cytokines are inflammatory driven or they help in wound healing. But there are proteins which are the building blocks of our system, and that's where you look at a mass spectrometer.
We can patch the mass spec.
Can you patch the mass spectrometer? It's very important because mass spectrometers, this is a mass spectrometer, very, very, it's the whole condition of how to keep this running. It is a very challenging machine. And it is at one given point of time, you can see thousands of proteins can be analyzed, and we are just going to upgrade our mass spectrometer to a higher one. So we call it as omics, O-M-I-C-S. Omics is metabolomics, lipidomics, then you have the proteomics and many other things. So it's like a mix of everything. So what happens is every gene, whatever the DNA, the mRNAs and all that has to do a work. Genes will not go and start working out for you. The genes has to secrete certain amount of proteins. I'm sitting with very accomplished researchers here, so I'm still, pardon me, I'm trying to make it very simple because there are a lot of youngsters here. So it's important that these proteins are the ones which are the messengers of the DNAs, and they produce, whether they produce tumors or they produce something which helps to recover, is driven by the genes. And what happens is identifying those proteins, we call it heat maps here, am I right?
It is.
And these heat maps have different signatures, and these heat maps, this is not, if there are proteins which can go into thousands and thousands of proteins. And when you have a heat map, there is a reference of how each protein is altered, and based on these proteins, you can identify what type of a healing process is happening.
Indeed.
And this is something which is important for doing a lot of research work, because, for example, I had a discussion today morning that there is my friend here from AIOS, he said that we have bring in a different type of a laser which causes least amount of wound disruption. I said wound disruption is not driven by your clinical data, because you will not be able to make out anything, because whether you create more burns or less burns, your next day outcomes will be 20 by 20 because you can't see. Of course, confocal is one of the ways, but the real way of how gentle you are on a tissue is driven by how your proteins behave or misbehave. So when you have a misbehavior of your proteins, then your whole alteration of your process is different. So it is very important for technologies of companies which bring in these kind of lasers, whether diabetic lasers or non-diabetic refractive lasers, to see how gentle you are on the surface. Yeah. And the whole process is driven by proteins, and there is a group of people who are working with us who also deal extensively on the signaling pathways. That means each of these proteins have a signal, and these signal, for example, there is a protein X which signals for it to do something. So in that heat map, you have to, from those thousands and thousands of heat maps, you have to find out what signal is driving and what kind of pathways. For example, in this is a driver. So what we try to do in our system is, we make the scientist to draw out this kind of a pathway, and you can, if you can, I don't know whether you can zoom in those small genes out there. Excellent. If you can zoom in more, like for example, you can zoom that MEX12, ERK1, and alpha SMAs, multiple of these pathways. These proteins are the ones which you can see, and those are the ones which can cause fibrosis. And it is important that when you look at these kind of proteins like alpha SMAs and all that, these are the ones, for example, you do a fantastic lasers on your retina or any of those surfaces, you may have end up in scarring. Sometimes you do any procedure, nothing, scar doesn't happen, or your PVR, so multiple things. This is on the keratocyte, this is from a cornea, but the same thing applies in many other parts of the system. So the whole idea of what we are doing, the mass spec out there, is to pick up these
novel proteins.
So when you see this kind of a pathway out there, you are actually seeing those proteins being picked up. And then once the proteins are picked up, then we start looking back at what and how do we actually address these proteins. So this is a very interesting concept of wound healing. Again, we need this. So if you can go to the holotomography ones.
Yeah.
These are holotomography experiments. We have if you can patch the holotomography.
Yeah.
These are something which is excellent demonstration of, I mean, if you go back to your MBBS or even your biology classes, you would see a cell having, your mitochondria and all those images. You can actually see all of them. Earlier, to create a desiccation stress, that means to create a dry eye environment, you had to create in a rabbit model. And I was told when the first restasis was used, they had to stitch the lids of a rat, and so that the rats can't blink, the mouse can't blink. So when you can't blink, you create a dry environment. It was so barbaric in trying to find out whether it works or not. For example, here these are corneal epithelial cell lines. You create a desiccation stress. You can see that the cells are under stress, and then you use, say, a cyclosporin, rifetigrast, or one of the drugs, and you can see how it works. Interestingly, you can also see how for example, how does the cyclosporin work on a scarring. TNF alpha is something which causes more of wound healing cytokine. And if you can enlarge that the patch there, so it will be interesting to see how we no, not that, the one from the osity of the cells.
This one.
Yeah. Okay, if you can make it bigger.
Funny. The NanoLive on full screen, please.
Those are called NanoLive imaging, and it has actually bypassed us working on animal experiments for many of these things, because now we can real-time work on real cells. We also have some retinal pigment cells. We have a very beautiful tumor, retinoblastoma cells. Swami.
Yeah, I'm going to show you.
So, and we can virtually look at any cell, and you can create some. This is how the 3D cell explorer works. Swami can now explain.
Yeah, it is a holotomography. What you see here in a chamber is where you place your cells in a dish, and this chamber is controlled with humidity and the gaseous exchange which you would have for your normal cell to grow. And you have an objective with the fluorescence light coming through the objective end, and what you see rotating is the mirror which captures the whole thing in all directions.
Can show the screen also.
Yeah.
What you see on the screen, live screen. Show the screen, please. What is the output of the screen?
Yes.
No, not yours. The screen of
the lab. Yes.
And this is what he's. No.
I think it's getting swapped. Can we go back to the NanoLive monitor?
Yeah.
Yes. It is capturing, if you can see, the blue bars.
And
you can zoom out. Praveen, can you zoom out? You can get the entire frame. Yeah. So every few seconds, a picture at enormous depth is taken so that you can have a 3D version of the entire cells.
We also have access to animal labs, and
Yeah.
we have some imaging from the animal labs. If you can show how the imaging of the animal labs is,
Yeah.
so that this is the work
Funny.
we do. And most of the animal related work is happening on the gene therapy part, and also on areas of
So
yeah. This is
So we capture these kind of images from the animals. This is coming from a mouse, and this is the mouse cornea as well, but I'll show you how exactly it is done. We have an entire imaging suite, and we also have the imaging facility. We can't go inside, so we have taken a video, and you can see these various cages. We have cataloged animals with different treatments, with different age groups, and they have their feeds and bedding always checked. We have a veterinarian who takes care of their health, and we have to do with according to the IACUC protocol. So
the ethics committee of animals is more stronger than
the human
em-
ethics committee.
Thanks to Menaka Gandhi. I think it's very, very difficult in our country to do anything which is even unethical on an animal model. In fact, it's very strong.
Yeah,
And I think the team and Tirumlesh is very good with animal work. He's very gentle, and he does a lot of animal related work in this.
and
Interestingly, we can also do an electroretinogram. I think Purna Chandra will start looking at ERGs on those animal models
yeah.
as we move on.
And this is somebody who is trying to get the anterior segment OCT live. The animal is alive, it's anesthetized, it's comfortable, it does not go through any kind of difficulty. And you can see the live imaging popping up there. And this is the live imaging of a posterior segment OCT, not from the same animal, from a different animal.
So we'll go to the gene therapy
Yeah.
unit. So that's something which is then the talk is also going to be followed by that. The gene therapy units have a very strict criteria in terms of who can enter, and because of the challenges of we work on gene modification. So there are different ways of how we are looking at gene therapies. It may look it is not too far away from reality, as I see by 2030, when we all be hopefully sitting in this place. I think lot of sessions will be dedicated into how therapies like this can change the treatment outcomes. So if you can go and patch the gene therapy units.
So we couldn't patch because
Lot of these
we are not allowed to get inside because government has given the
Okay.
thing. So we have taken videos,
But that I think Pooja is anyway going to speak about the
Right.
corneal part of it. But
and
I
these
mean,
are,
can just discuss this a bit. I think this is what Tirumalesh showed. Those
and we have a video.
you can explain.
So these are the various in vivo models which we have. Finally, the gene therapy product, the medicine, is going to go inside these animals. So we have to set that up. The AAV production, that is the vehicle which is going to carry the good gene, is produced in-house in the lab, so to make it affordable. So we have to make millions of it, vehicles. We call it vectors. Adeno-associated vectors are the vehicles, which is the most important part of the genetic gene therapy or the genetic medicine to begin with. So we can't just make it. We have to purify it to a level where we have to be absolutely sure that only the,
and its development requires a lot of
That's our
scientific
director.
involvement from a variety of different disciplines that includes molecular biology, genetics, biochemistry, animal sciences, vector technology, and finally, the know-how to actually make clinical grade vectors. That's what we have achieved here in environmental health, and the future is now.
So we definitely have come very close to it. Because of indigenous development, the cost is going to be a lot more affordable. Start to finish happens in one place.
Can you also ask somebody to patch up about storing sample? A lot of people
Yeah,
ask me
biorepository. Yeah.
that how do we store samples? What is the whole protocol of storing a sample? Because that is what helped us to reach here. 20 years back, I give my credit to Dr. Kumar Menikavel sir when he joined us as an advisor. The first thing he said was, don't throw
Biorepository.
anything. That time we didn't have any lab, and we didn't even have this. In fact, I thought this costs a lot. In fact, our slit lamp costs a good slit lamp costs less than this.
Yeah.
So I didn't realize this, but we were storing in Narayan Rudyalaya blood bank, and along with one slit, we had kept 5 years of thing. They threw it one day without
Yeah.
without our
Telling
notice.
us. Yeah.
Then we realized. So this hardly costs anything for people starting. This is like a like a Bank of Reserve Bank of India or Bank of London thing, and this houses thousands and thousands of samples. A lot of my ex-fellows and colleagues are here. Whatever they collected is all here and so well categorized. There are more than 30,000 odd diabetic samples.
Yeah.
We have close around
We crossed 80,000 to 1 lakh, sir.
yeah, and 1 lakh odd samples of everything. So and the question is, what do we do with this? See, out of those 80,000, there are thousands and thousands of pathways, thousands of new diseases, thousands of new pathways yet to be discovered. So biggest challenges is, for example, you can see that every corridor in our research, the 3 floors of research, every corridor has we only started with 1.
Yeah.
And now every 6 months we have to keep buying it. And how much does one of those storage house? How much does it house?
More than 1,000. More than 1,000.
And ask one of them to just open up one of the box. This is for a lot of young
Biorepository,
people
can you
or
open one of the box
institutes
which you
wants
just
to
opened,
get
please?
into this. My only advice is don't throw precious samples. You're doing a cataract on a RP patient, or you're doing anything which is interesting. Even if you don't want to buy, please find a place where they can house it for you. And if you can just go close up and just show how it is, it's like very well, each of those samples may contain a tear film, vitreous, aqueous, trabecular meshwork, sometimes even the CSF from.
Can you pull up one of the tubes, please?
This is gold. Kumar, even today, he says this is for a scientist. This is gold. How you keep it in your bank. Can you go close up and see each one is labeled. Each one has a name, a number, and if you can, we don't have a QR system yet. Yeah. But if you type this number, you'll get the complete patient history, diagnosis. I have at least found.
You can close it.
At least 2 important genes in refractive surgery because we had it, and what happened was we identified a novel mutation of lysyl oxidase in refractive surgery, and that became a big area of interest, and now that is the area of that because we had lenticule preserved in it. So housing your samples is very important, and I think that is the first thing which the researchers should do.
May I add something, sir? When I first joined RNA around 2015, 10 years ago, something very unique was happening. So they were all going through this biorepository to find out a batch of patients developed post-PRK haze, and you've been collecting the epithelium as part of the PRK and you've been storing it. Around the same time, they were able to see that this is before haze, okay, during PRK. A very unique signature was identified, and PREX1 turned out to be an indicator of PRK-induced haze predisposition. So that came in because of the biorepository samples. That was my first experience in GrowLab, actually, on how translation can be.
Can you patch in people working on corneal haze?
Yeah.
And
yes.
And also people, Sangwan sir is here. I think we have not been able to achieve his level of thing in regenerative stems,
yeah.
At least somewhere close by with his inspiration. If you can just show the work happening on
Western blotting and qPCR.
Yeah, and there is a team also working on biodiffusion. That means if
we have it, sir.
So biodiffusion is an area of interest where if you give that LFA1 antagonist, which Tirumale said, there is an eye drops on it. You know, I was telling them that if an eye drop, we need to know how much of that eye drop actually reaches the retina.
Biodiffusion.
And that is a whole work of biodiffusion. And if you give, say, people say I give an omega-3 or a resveratrol, how much of it actually goes to one retinal cell? And there are various ways to look at these chromatography and those kind of work. And this is a work of biodiffusion. That this is something which is very important, because you know sometimes we don't know how much whatever we eat, say an omega-3 fatty acids, how much of it actually comes to the corneal epithelial cells. And this is
That is Ravikran, he does the biodiffusion work. We
And
can zoom out and show the
that's
HPLC.
okay. That's okay. In the interest of time, we can move to the haze work, corneal haze.
So,
And there was a discussion with the Heidelberg friend from Heidelberg. We were telling that, you know, we can look at what are the haze forming, what are the surrogate markers in the epithelium, and how this can have an impact in the hidden immunology. And this is the qPCR work which
where
we did.
we do both haze work as well as ectasia work.
Oh.
So we can patch in qPCR and Western blot.
Can make it a little bigger. No, not that. The
The
guy
video,
on the other
yeah.
side.
qPCR and Western blot, please. Yeah, we can focus on the live. We can take the slides off.
Make it live, and the last one would be on iPSC,
iPSCs, yes.
pluripotent stem cells.
That's right.
And this is the work where you can see those graphs when you're studying those multiple genes. And
It goes off again. And Western blot also, if you can show. So what you see is a quantitative PCR machine, which is the quickest way. Now, because of COVID, everybody knows what PCRs are, but we're using it for epithelium stroma, any tissue which you want to have a quick breakthrough in the molecular pathway, which gives you the complication or a unique presentation.
So we move to the iPSC cells,
Yeah.
and we can end this session. That's okay. Is Kaushik around?
Kaushik is there, but I took all his videos.
So just quickly
Yeah.
show, just to complete it.
Kaushik, if you're there, please connect. Yeah. So any cell to any cell is what he does. He can take, you give him a corneoscleral rim or a blood, he can convert this to corneal epithelial cells, endothelial cells, retinal pigment epithelial cells, RGCs, neurons, whatnot. He's a magician there. And he's following a very important Nobel Prize winning method or concept called induced pluripotent stem cells. He takes the somatic cells, brings it to stem cell state, and pushes it to this type of cell you want to make. Functional indeed. He wants to do transplantation as well. So what you can see here is from blood, he has made keratocytes, he has made corneal organoids, he and his team, I should say. And he has made corneal endothelial cells, which is ready for transplanting for some of the dystrophies. And
interesting thing is about this is organoids are creating virtual cells.
yeah,
You can create an organoid from melanoma or retinoblastoma. So instead of trying multiple drugs on creating a
In
model
animals.
on animals, you take that whole organoid, grow the cells from the same, say, retinoblastoma or melanoma, any cell lines, and start try different drugs. Kagan was working on
yes,
it.
that's right.
And then see which one of these drugs works on this. So
And then-
organoid-based research to create models is a fantastic model of human diseases. I
Yes,
think
disease modeling as well as for pharmaceutical screening before we take it to the animals and further. And he has made a beating cardiomyocyte from a corneoscleral fibroblast. And that is a skeletal muscle which he has made, he and his team. All of these are for functional studies. If you can't get a primary cardiomyocyte, you can't get a primary skeletal muscle from a patient. So we use this technology. And he and his team has got neurons for us to interrogate some of the neurophysiology work.
beautifully you see how they connect to each cells. And that's how a thought process is done. Thank you, Swami.
Yeah.
Thank you. Thanks a lot.
Thank you.
My great honor to call upon Dr. Sangwan sir for his talk on translation research in ocular surface. So, any quick comments from both of you, sir? No, Professor Sundareshan and Kumar Manikavil.
I think right now the current trend is gene editing. So you can refer the what is the current trend in the field of genetics or gene therapy is a kind of gene editing. Using the CRISPR-Cas9, you can able to change even a single base pair. So that's what Narayan Nethralaya is doing. Wonderful job for different strategies. There is an article in the one of the highest journal in the field of ophthalmology that is called preventive medicine and progressive retina. Thank you, sir. So that is the 29 impact. 29 impact in that they given what are the current genetic strategies for gene therapy, even in the early stage or end stage. End stage in the sense you have to adopt the optic genetic technology like that. So I think right now we are in a right stage to reach the how to change the research from bench to bedside. I think many labs in India they are taking initiative to transfer the technology from bench to bedside. That is some comment. That's what we are seeing a lot of improvement from the Narayan Nethralaya labs.
I have some comments to make, but I'll do that once Virender finishes.
Thank
Please
you.
go ahead, sir.
Thank you very much. After those fascinating videos and direct, live transmission from the lab, it's very energizing that such a high quality work happening in our in some of our labs in country. I'll share briefly about cell-based approaches for ocular surface translational research. I have no commercial interest to declare. Here is a 9-year-old boy brought by his father from Maharashtra. He had this appearance in the left eye following a accidental Chunna injury, and the boy was brought up because he wanted the eye to be removed. He was not able to go to school, and the father said, please doctor, remove this eye, because many doctors told that nothing can be done about it. What we did was a technique called simple limbal epithelial transplantation, in which we take small limbal biopsy, cut into pieces, clean up the ocular surface, and this is what you can get. So to achieve these kind of results, we were able to do because we were doing from 2001 the cell-based cultivated limbal epithelial transplantation, which means we are harvesting the limbal biopsies. From there, we are growing the cells for 2 weeks diligently, and then we are transplanting in human affected corneas. And this is an example of a patient who has 22-year follow-up of a autologous cultivated limbal epithelial transplant followed by a PK, and she is now a grown-up lady, and when she had this injury, she was about 10, 12-year-old. When we have such a cornea, most of the doctors about 10 years back used to think that nothing can be done about it. When we did a slit in this patient in 2011, you can see there is some scarring, and at that stage we used to think, okay, we need to do a DALK or a PK, and we offered that to this patient in 2012. But he said, doctor, I have enough surgery, I don't want. So he continued to follow up, and to our surprise, cornea continued to clear. And in 2016, he improves to 20/40. And why and how this happened? This, we thought, when we were growing the cells in the lab, we realized that there are mesenchymal cells in the cultures, and when we compared the gene markers of, gene signature of those cells and bone marrow derived mesenchymal cells, they are very similar. Based on that idea, and based on the work of Professor Fundenberg from UPMC, Pittsburgh, we started working on the idea that, okay, mesenchymal cells can be a solution for many of the corneal disease. And why we think that way? Because if you're a cell biologist, you will look at, well, there is a narrow window between a fibroblast and a myofibroblast. And there are various factors working on it, microenvironment and the cytokines, to develop into a transparent cornea or a scarred pathological cornea. The work of Sunil Chauhan from Harvard Medical School and Pittsburgh and our own work made us understand that these mesenchymal cells are very important cells in the body and in all the organs, because it can help improve the microenvironment, it can support the progenitor tissue specific progenitor cells, and it can immunomodulate. So based on this work, Sunil Chauhan's group has worked out that HGF is the key part of the secretions or cytokines, and based on that, they have a neurotrophic keratitis clinical trial, which is now done by a company called Claris Bio, and that is a startup. And I think the mesenchymal cells are answer to many of the treatments or the disease that we see. For example, a patient with Chuna injury, we can treat him once we remove the Chuna and use the mesenchymal cells which are grown, or maybe exosomes derived from the mesenchymal cells. These exosomes, we are working very closely with a company called Pandorum, which is a startup from Bangalore, and we are able to develop tunable exosomes. Exosomes are small vesicles, nanovesicles, which contain all kinds of cytokines, growth factors, and they can be very tissue specific. So I'll give you an example of that tissue specificity, that we are developing a product called Curogenics, which is liquid cornea essentially, and we are developing for neurotrophic keratitis. And this is a rabbit model where we have published extensively and shown that it is quite effective. And this is the paper we recently published, that Curagel, which is a hydrogel functionalized gelatin and hyaluronic acid, along with the exosomes derived from the mesenchymal cells. And this is, the beauty of this product would be that you use a drop and show the microscope light for 30-40 seconds, and it polymerizes and becomes a solid in place. Corneal epithelium grows over it, and it gets integrated. And this is an example of a rabbit surgery that we are doing. When the scar is created, then you remove the scar and fill that crater with the liquid gel, and leave for 30 seconds, solidifies, and the corneal epithelium will grow over it, and that's it. So what we are thinking that, this is a human scar, the same video clip. So now if we have to treat this patient, either you have to do a DALK or you have to do a PK, but most clinicians say this is scar not very big, maybe not a good idea to do surgery. But once you have the liquid cornea, you will say, yes, we will treat it, we'll remove the scar and then fill the crater with the liquid cornea part. Similarly, when you have keratitis being treated, whatever the type of keratitis, they reach a stage of neurotrophic stage. At that stage, you can introduce this liquid cornea with exosomes, and that can restore the transparency. Similarly, I think this kind of treatment in fungal keratitis, when you have early fungal keratitis, instead of using for many, many weeks and months topical antifungals, maybe I can remove this infected area and fill the crater with the liquid cornea. So I think this would change how we look at the corneal diseases. Similarly, when you have bacterial keratitis or infectious keratitis, when the infection is healed, you have PED or you have neurotrophic element. At that stage, you can start treating with exosomes or combined with the gel. So when we do translational research, and what Rohit and his group were showing from the GROW lab is basically identifying a potential treatment, then do your preclinical studies, animal models. That's what I just showed you, and that's what we are doing. And then you move on to, once they prove the safety and efficacy, you move to human trials. And currently at that lab in SCH, we are working on Stevens-Johnson syndrome, the molecular basis, VKC. We think that mesenchymal cells or exosome-based treatment is possible. Similarly, we are working on OSSN and corneal fibrosis and chemical and thermal burns. These are the markers, the different students working on these disease models that I described. So in summary, cell-based approaches are key for the development of new treatment for cornea and ocular surface. And I go back, when I started corneal cultivated limbal epithelial transplant in 2000, 2001, that led to SLET, and when we're doing the bioengineered cornea, that led to the concept of liquid cornea. So, translational research accelerates novel therapies for ocular surface disease, bridging molecular insights with clinical applications, and future prospects of personalized medicine and advanced nanotherapeutics can be fulfilled. Thank you very much for the kind audience.
Thank you, Dr. Sangwan. We'll be moving to the next talk by Dr. Pooja Kumar on genetics and gene therapy in clinics. And when she's setting it up, I'll make some comment. So the field of medicine started picking up speed and relevance in the 19th century, where we're doing clinical examination of examination and palpation, auscultation, and so on, right? So, but then in the last century, in the 20th century, what happened is that we moved into preventive medicine, where we knew the target groups which are susceptible for a particular disease, and then we screen them before they start to get the disease so that we can prevent. But this century is going to be the field of omics, as Dr. Shetty mentioned, from genomics to phenomics, and you have so many other omics in between. And today with genomics and other omics, you can do a predictive medicine. Okay, that's the field in which it is. If, for example, I'm going to develop AMD in 5 years from now, and then an ophthalmologist is going to treat me. But today, there are a lot of molecular changes that are happening in my retina, and that's going to come in my tears, and that's going to get into my blood. So you as a clinician today, if you can pick that up and then tell me, look, you're going to develop AMD in 5 years, and then you start to manage me, that's the way the field of medicine is going to move. So you as clinicians will be predicting diseases more than actually diagnosing them. Okay, so that's the major shift that's going to happen, paradigm shift. And all of us have to do research for that, and therefore two sides of a coin. One side will be the clinician, the other side will be the basic science scientist. So with that, I hand over it to Pooja. Go ahead.
Thank you, sir, and thank you for making me a part of this wonderful session where we talk about the translational science and how it's going to change our practice towards customized medicine. Now, we all know that keratoconus is a progressive corneal disease characterized by corneal steepening and thinning. But what happens is that it leads to blur vision due to irregular astigmatism, leading to defective stereopsis, and it does affect the quality of life, and it has an emotional impact on patients having this. Now, we do have standardized gold standard treatments for like corneal cross-linking for early stages of the disease and corneal transplant for advanced stage of disease. However, we do come across patients who are extremely advanced, who probably need a transplant, and at times when you have done a lamellar keratoplasty or a penetrating keratoplasty, the transplants do fail, and if you have done a cross-linking, then there is still a progression of the disease in spite of doing a cross-linking as well. Now, we do know that IRA being an inflammations do play a good role in the keratoconus disease, but there is a genetic aspect as well in the disease which are beyond your cross-linking and transplants. So the role of genetics in keratoconus are, as Dr. Rohit mentioned, that when he started storing the samples did help in identifying two very important genes, and one of the very important genes is lysyl oxidase. And the work started back in 2010, and now we are in 2025 looking at gene therapy. Lysyl oxidase is nothing but you can see this pink dots here. It basically holds your collagen into place, but if there is a lysyl oxidase deficiency, the collagen bundle starts splitting away, and that's what leads to the disease which is known as keratoconus, and whenever it's lesser, it leads to progression of the disease. Now, this was the first paper published where they did find out that with the increasing grades of keratoconus, the lysyl oxidase levels are lower. And when they did, because keratoconus comes across as a localized disease, the lysyl oxidase was measured separately from the cone area and the peripheral area, where they found that the lysyl oxidase was lower in the cone area and the periphery was healthy. So that does prove that it's a localized disease, and lysyl oxidase does have a role to play. And if the lysyl oxidase was on the lower side, where you can see here, those patients had a higher risk of progression of the disease after cross-linking as well. And the cross-linking technically failed in patients with lysyl oxidase being on the lower side compared to that of being on the normal side. So again, showing that even if the lysyl oxidase is, even if you do a cross-linking, but if the lysyl oxidase is lower, the patient is at high risk of progression. And not just that, in a very healthy individual which you subject for LASIK or smile surgery or any refractive surgery, sometimes you don't have an answer of why these patients are progressing. Again, this was the paper published in 2018 where we had saved the lenticule. You saw the biorepository where we save all the samples. Now this patient was extremely healthy and developed a post-refractive surgery, a post-smile ectasia. We had preserved the lenticule of this patient, so when the patient developed ectasia, we went back to our biorepository, took the lenticule out and analyzed it with the gene expression analysis, the qPCR. And what we found was, again, that the lysyl oxidase in the ectatic eye, the more ectatic eye, which is the left eye, was lower compared to the right eye, which was an evolving ectatic eye compared to the controls. So it showed us the power of preserving a lenticule in 2018 when the patient underwent surgery. 2023, when the patient developed ectasia, we took the lenticule out and found out the defect. So again, not just keratoconus patients, but a post-refractive surgery ectasia patient can also have a low lysyl oxidase level, which can lead to ectasia, is what was told. And we also, as mentioned before, we know that sometimes a lamellar keratoplasty can fail, and we don't know the answer. So the answer lies in the genetics, and if there is a genetic defect, we do need a gene therapy, and an ideal gene therapy would be something which is safe and non-immunogenic introduction of genetic material by an able vector into the target the cells, rendering them to capable of repair, replace, or overpowering the functions of the defective or non-functional genes for therapeutic purpose. Now, there can be three types of approaches when it comes to gene therapy. It can be gene activation, inactivation, or silencing of the mutated gene, or you can correct the genetic defect by replacing the mutated gene, or you can have an addition of a healthy copy of the gene that will ectopically express the therapeutic protein. Basically, it will overexpress the ectopic disease protein. So you can have various types of gene therapy when it comes to any corneal disease or any ocular disease. So this is our Center of Genetic and Gene Therapy at Narayan Nethralaya, along with the GROW lab, and this is a dedicated clinical grade gene therapy vector production facility, what you saw with Dr. Swami. And this is, you have to wear specifically a spacesuit, enter inside, because it's a class 100 facility where you have to have the cleanliness at a level of class 100. So even a minute particle which you see in a microscope is not allowed, and that's why you have to take a shower, wear the spacesuit, go inside, and then develop this viral vectors which can be used for injection. So this is what we've developed a few years back, and as right now he mentioned, because of the regulations for approvals, the government has asked us to keep it clean for a few days. So what are we aiming with the gene therapy lab? We are trying to develop AAV-based vectors. And why AAV-based vectors? These are the viral vectors which have an efficient delivery of genes to both the dividing and non-dividing cells, which helps us to have high levels of gene expression. Because they are viral vectors, there is absence of viral genes for an basically trying to avoid an undesirable immune reaction, because the biggest fear with gene therapy is that it's going to lead to toxicity or a reaction or a rejection. So viral vectors are quite safer in that ways, and it helps us to have a lesser toxicity reaction even if it's going to be there. And viral vector is going to have a long-lasting expression based on this because of the way it's been mechanized. So these are the few studies which we have done for lysyl oxidase. This was done on the ex vivo cornea model, where one eye was treated with a GFP, which is basically a controlled viral vector, and lysyl oxidase, which is the viral vector which I mentioned. And when you inject the corneas of the mice, you can see that the expression of lysyl oxidase is increasing compared to that of the control eye. Not just that, I did mention in the beginning, if the lysyl oxidase is lower, the collagen starts slipping away. It's also increasing the expression of collagen 1 and 4 bundles. But the best thing what it's doing is, again, the keratoconus is inflammatory in nature, as you heard, and it's been proven. The lysyl oxidase is reducing the inflammation level. So it's like a double benefit. It increases the lysyl oxidase, stabilizes your cornea, and reduces the inflammation. And this was also seen in the lenticules which were preserved in healthy eyes, if it works in the SMILE lenticules as well, and which is doing that. Now, because the collagen fibers are slipping away and there is ectatic disorder, when we inject lysyl oxidase, you can see this white gaps are basically closing here. That means it's kind of resynthesizing the collagen. So it's again a good thing what we see here. And when there is an increasing expression of lysyl oxidase, the collagen distribution is increasing, whether the strength is increasing or not. So we did atomic force microscopy, and you can see that where the eyes where we have injected lysyl oxidase, the elastic modulus is also significantly increasing in these eyes. So the next step before we go into patient trials, we were asked to do in the rabbit eye corneas, the gene therapy and see how it's acting. So you can see the slit lamp microscopy. The corneas are clear at 12 weeks post-injection. OCT, you can see that there is no thinning of the corneas, but it's being maintained. There is no side effect. The fundus and the OCT of the OCT also shows, they don't have macula, but OCT also shows to be normal. There is the specular microscopy also is within normal limits, basically no damage on the endothelium. And this is a confocal microscopy suggesting that that it's not causing any keratocite activation or myofibroblast formation, so not causing any scar, and the corneal tensile strength is also increasing. So this is what has been our journey. Now, why cornea? Because you have imaging modalities like topography, biomechanics, OCTs to measure whether they are working or not, so it is easier for us to do a gene therapy in the cornea to begin with. A lot of children nowadays, especially because of allergies, are undergoing many of these emotions because of keratoconus and trying to probably have going into a depression, what you can say, and it's very difficult to bring a child out of depression. So if you can have a gene therapy for these patients, you can actually create a good psychosocial impact for these diseases. We are not just limiting to keratoconus, but we are also, as Dr. Rohit mentioned and Dr. Swami mentioned, that there is a PRK haze which is involved. So gene therapy can also be used for treating corneal fibrosis and angiogenesis, which is work in progress for our team, and this is what we have done. And you will need a genetic testing to identify whether there is a genetic defect or not. And there are multiple gene tests, especially the next generation sequencing, which will help us doing that. Thank you.
Thank you, Pooja. As Pooja mentioned, this is one of the state-of-the-art lab in the country, the gene therapy lab that is being run by Dr. Kaghosh and well supported by Dr. Thirumalesh with his animal and clinical experiments. So thank you, and we'll have questions at the end. We'll try to finish the session as I discussed with Dr. Sangwan and Dr. Dhanashree. So we'll go ahead with the next talk by Dr. Dhanashree Ratra. She's from Shankar Nethralaya, and she'll talk on the macrophages at the retina in diabetic retinopathy. Dr. Ratra.
Good evening, everybody. Thank you at the outset. Thank you for making me part of this wonderful session. So I'm going to talk to you about macrophage-like cells in the retina in diabetic macular edema. So no financial disclosures. So with the advent of optical coherence tomography, angiography, these macrophage-like cells have been shown to exist in the vitreoretinal interface over the internal limiting membrane in the retina. And these have been identified as retinal microglia. Now we know that these microglia are the innate immune cells which exist in all the layers of the retina. And basically they play a big role in the maintaining homeostasis in the retina. So microglia, when in the resting state, they are pro-survival, but when they are activated by some stimuli, they produce a lot of cytokines which can be neurotoxic. So this microglial activation is a common hallmark of many retinal diseases. And the opportunity to visualize these cells and study their behavior in vivo is unique, it can give us unique insights in the pathogenesis of several retinal diseases. And so this is a image showing you the phases in the microglial activity. So you have a resting stage in homeostasis, then you have the activation stage with the activation phase with the stimuli, and then they migrate to the area of the stimuli, the insult, and they become amoeboid, and they lead to phagocytosis, thus maintaining the homeostasis. So in order to visualize these MLCs in vivo, we take a OCTA scan, the 6 by 6 millimeter scan centered on the fovea using the Zeiss Cirrus 6000 machine with a good signal strength. And then the scan is manually segmented in the vitreoretinal interface at a slab 3 microns above the internal limiting membrane. And then this slab is exported to the ImageJ software for further analysis. Now, a stepwise semi-automated binarization protocol is used to extract and visualize this MLC macrophage-like cells. And here you see this in the last image. So these are the macrophage-like cells. So these are the cells actually, but very difficult to visualize them. So we have colored them red and then superimpose them on the angio image for better localization and topographic mapping of these cells. So so let us see what happens to these MLCs in diabetic retinopathy and diabetic macular edema. So we did a pilot study of 86 eyes of 86 patients and grouped them into eyes which had no diabetic retinopathy versus eyes with diabetic macular edema. And what we saw that the MLC activation was seen at distinct stages of diabetic retinopathy. The MLC density as well as the count increased with increasing severity of diabetic retinopathy. And nearly about 2 to 3.8 times increase was seen in these MLCs in cases of diabetic macular edema. And it was, again, it was correlated with the severity of the diabetic macular edema. The MLCs were seen clustered along the blood vessels, microaneurysms, and ischemic areas. And they were also significantly correlated with the glycosylated hemoglobin values. So these images, they show you the left side show the MLCs in the diabetic retinopathy, where you see very minimal images, and they are increased in the diabetic macular edema. So possibly the MLCs can act as biomarkers for disease progression in diabetic retinopathy and diabetic macular edema. So central foveal thickness and visual acuity are traditionally the two biomarkers which are used to measure the disease activity as well as the treatment response in diabetic macular edema. And what we saw that these MLCs showed significant correlation with both these biomarkers, indirectly telling that MLCs also can act as biomarkers for disease activity and possibly treatment response. So these images, they show that there is an increase in the MLCs in patients who did not undergo treatment for DME. At the follow-up visit, you can see much more MLCs in the patients who did not have treatment, whereas patients who underwent treatment and showed good treatment response, at the follow-up visit, you see there is a reduction in the MLCs. And this table just shows us that there is a significant difference in the MLCs in patients who underwent treatment from pre-treatment to after treatment. So can MLCs act as biomarkers for the DME treatment? To understand this, we did a regression analysis using MLCs and correlated them with various biological markers. We included several predictors such as lipid-related measures, that HDL, etc., then liver function test, hematological parameters, glucose tolerance test and blood sugar, oxidative stress markers, growth factors such as VEGF, basic FGF and PDGF, and inflammatory cytokines. We had a robust regression model with a very good significance level and association. And what we saw, surprisingly, that the VEGF was negatively associated with the macrophage-like cells. So what it means that when the VEGF macrophage-like cells increase, the VEGF level was seen to be reduced. And this indicates that in eyes with increased MLCs, the anti-VEGF driven response may be likely to be poor, because obviously there is no VEGF, so the anti-VEGF response might be poor. And these are probably the eyes which we call them as non-responders, and these are the eyes which we keep treating with anti-VEGF but without any response. And possibly they might be more candidates for intravitreal steroid treatment rather than anti-VEGF. And this finding was actually corroborated by a clinical study by Rangwani et al., where in the retinal vein occlusion study, they found the increased MLCs were associated with thicker maculas, they were difficult to treat, they required more injections of anti-VEGF, and they had worse visual outcome. So this possibly indicates that the VEGF and MLCs, they are negative correlation. So how early does the MLC activation occur in diabetic retinopathy? So this is a graph from the neurological study, and in neurological studies such as Parkinson's and Alzheimer's, it has been shown that the MLC activation precedes clinical diagnosis of these disorders by nearly 10 to 15 years. So whether the similar thing is seen in diabetes was our hypothesis. We know that there is a state of pre-diabetes which exists nearly 3 to 5 years before the clinical of diagnosis of diabetes and nearly about 15 to 20 years before diabetic retinopathy. In an earlier study on pre-diabetes, we showed that there is a early neurodegeneration seen in pre-diabetes eyes without any overt structural changes. That means without any vascular changes or neurological changes, the functional changes were seen. Now in this study we looked at again MLCs in pre-diabetes and what we saw that the MLCs they did show excellent correlation with the multifocal ERG amplitude. They also had reduced foveal thickness. Although there was no structural changes, that means there were no structural changes in ganglion cell layer or RNFL, and there were no changes, vascular changes at all, but these early neurodysfunctional evidence was there. Apart from that, the multifocal ERG showed also topographic correlation with MLCs. That means the points of where the multifocal ERG amplitudes were reduced showed increased MLCs. So the MLCs, they not only predict neurodysfunction, but they are likely to even tell us the sites of future vascular changes, because in diabetic retinopathy it was seen that these areas which show reduced MFERG amplitudes will develop vascular changes about 3 to 5 years later. And here in pre-diabetes we are actually possibly predicting these sites nearly 15 years before the clinical changes can occur. So possibly MLCs can be precursor for these vascular changes. So to conclude, the non-invasive, label-free, in vivo visualization of the MLCs can help us understand the pathogenesis behind several retinal diseases, and MLCs can possibly act as disease biomarkers for diabetic retinopathy, as they show excellent correlation with the severity of diabetic retinopathy as well as visual acuity and central foveal thickness. The MLC showed negative correlation with VEGF. This of course needs further exploration, but it can help us explain the non-response that we see in certain patients. The activation of MLCs occurs at very early stage, even at the stage of pre-diabetes, which suggests that possibly early neurodegeneration occurs in pre-diabetes, and this possibly means that if we do some early neuroprotection, we might save some of the eyes from losing vision. So thank you very much for your attention.
Thank you, Dr. Atra. So we have two more people to present. One is Dr. Radhika Tandon, another is Dr. Sundaresan. So Sundaresan will go first and then Dr. Tandon will go. She's still setting up her slides. In the meantime, Dr. Sangwan has to move to another hall, so we'll have some discussion before these two talks. Madam, you can come. So we'll start off the discussion. I would like one of the first panelists, sir, you can give your views, and I have Purna also to give a view. Go ahead.
As clinicians, now we are getting into labs, and we have seen Dr. Tirumalesh doing great job of giving injections in the rats, and a lot of things are happening in the lab which we are weighted the molecular level, and a lot of things specification other than VEGFs, like Dr. Rohit had said. Other molecules have also been tried for the retina, which comes out well in most of the early studies. So great work by all the stalwarts, and liquid cornea by Dr. Sangwan, and macrophage by Dr. Atra. It's great to hear from the heroes of genetics and lab.
Yeah, thank you, sir. Please go ahead, sir.
I have a question to you, Dr. Sen. You used hydrogel as
So it's one.
that is a non-biologic.
Yeah.
Have you seen any rejections or vascularization?
No, not because these are studies in the rabbit, and we have maximum follow-up 3 months, and only few rabbits we have 9-month follow-up. So during that we don't see any rejection or at all. And hyaluronic acid anyway is used in ophthalmology extensively for variety of other applications.
Do you have experience with collagen biologics, collagen type 1?
Yes, we had, no, not collagen 1, but we had used recombinant human collagen, RCH3, for making the bioengineered cornea, transplanted in human, and we have done the pig and rabbit work in 2013 and 2014.
Apparently, we can do C3 around that also.
Yes.
To strengthen the.
Yes, of course. Thank you.
Thank you. So I have a very tangential question for Dr. Sangwan, Dr. Tandon, and also Dr. Atra. How to motivate the fellows and clinicians to do research, particularly molecular research, with their clinical knowledge and application? Dr. Sangwan, you can go first, sir.
It's a very challenging question, not only in India, I think globally, that the clinical translational researchers are declining, and in our country we don't have enough, or we never had a lot of people who have interest. So I think the only way we can motivate, inspire is by having a role model or examples that this is how you can make a difference. And if we have examples from different parts of the country, then it might change, but it's going to be a very slow process. Even like in LVP, when as there, not more than 10% people would be genuinely interested. Others, they publish because they want to get promotion or they want to travel. And most places that's the story. Research definition is different, like, okay, if you publish retrospective, if you do a case series or do a clinical trial, that's good enough. But getting interested in basic science, molecular level, I think that's the key.
Thank you, sir. Actually, labs are there, which is the most difficult thing, good scientist and good lab. So collaborating with such kind of places like Narayan Netralaya, Shankar Netralaya, Arvind Ali Prasad, AIMS, and of course with Dr. Sangwan's institution. So you can still do research. It's not that you need a high-tech lab that should be in the next door that you can do research, but you can still collaborate with people of this nature. So with that, I will ask Dr. Tandon to make her comment about how to motivate young clinicians into research.
Yeah, there are many reasons for the hesitation. So if we look into that also, it will help us find the solution. One is, of course, that the career path for science in terms of funding is still a hard journey. And where now all the people who are doing ophthalmology, they're having a lot of frontiers and horizons in going for clinical service and earning a very, very good salary. So that's one thing. Whereas the science journey is hard. And earlier, the tendency was to go for academic positions. Again, those have become very complicated. In the government system, often they're delayed, and then often they'll keep changing the sort of policies and what kind of salary structure would be there. Even in the medical colleges, there's a new system which is without a pension. So all these are making people go away from the path, discouraging people from going for the path of going for pure academics, because they see around them that after so many years of struggle, they have reached the stage and then they have to struggle again. So I think the key is that we have to make the environment facilitatory, and as you said, there are people in the labs who are basic scientists who require that clinical perspective, support, and they already have it, the lab, and they're already doing it, and they have some funding. So we need to have a network of known institutions. You mentioned a few, and we could have a few more, and then we can encourage people to work together, and and we have all the talent in our country. We do not need to go outside at all. There are so many people here who have the talent, they have the wherewithal. So we just need to reach out and learn how to work together. I think it will happen.
Yeah. My comment is that we should have a regular interaction, a meeting between clinicians and the non-clinicians. Because what happens is that even if we have a big academic institution, there is always clinicians are separate and the lab people are separate. So we do not know what the laboratory the scientists are doing, and the scientists have no idea what is happening in the clinical scenario. So unless there is a interaction and a regular brainstorming kind of a thing, meetings are done, it will not generate interest. So for the clinicians to have interest in that, I think some interaction is a must. So maybe a weekly or even monthly meeting should be held where you can, you know, toss our ideas around, and then only you will get interested people to do further, because this requires a lot of patience. It's not very easy to do basic science research. So unless they are interested and motivated, they will not do it. So I guess that is how we should go ahead.
Can I make a comment?
Yes, sir.
Follow-up comment?
Please go ahead.
See, I think the clinician think that to do basic science research, you have to give up your clinics and surgery. I think that's not required. I mean, it's just that you find little extra time or little time that you build your interest. For example, I have never had any extra time during last 25, 30 years. I always do 2 OPDs, very busy, and 2 OTs, 4 days a week clinical work. So I think it's more about your interest and finding that time, and think authentically believe that basic science can improve the clinical care.
Practice, yeah. That's a very good point raised. So, see, being a clinician, you know, we're all quite fortunate to get into medicine and then practice it. But research is something very important. So what your, you know, discipline in medicine gives you, we have to give it back to the field. Okay, practicing it, of course, curing patients, all those are very important. With that, we have these questions open to the audience, and I'll start with Purna, Dr. Purna Chandra, and then any question that you want to ask the speakers who made presentation, or with the Grow Lab, or any other thing that you want to raise and find out how can I do research. Say I'm sitting in a remote place, or I'm sitting in an institution which doesn't have a lab. So all these kind of questions, if you want to ask, we'll be happy to address. So we'll start with Dr. Purna Chandra. Yes, sir.
So thank you so much. I think most of the points you people have, you know, elegantly brought out and discussed. So something what I would like to add is, when it comes to, you know, treating disease, as we as a doctor are not limiting to the being retina specialist, ophthalmologist, anything, the toughest situation what we face is when we have to say no treatment. Patient comes, you have treatment, you do surgery, give some medicines, very easy situation. The challenges when comes when there is no treatment. And most of us are trying to chase this disease to offer some form of, you know, treatment. And the second thing, as Dhanashree ma'am rightly mentioned, and also Dr. Kumar Manikwalam sir mentioned, the future is all about predicting. Okay, now I am like this, what's going to happen after 5 years? So that is what I wanted to add on here. And regarding the gene therapy aspect of it, I mean, of course, that is my area of interest, especially the Stargardt disease. Pooja has nicely covered the cornea aspect of it. And Thiru is also working a lot on age-related macular degeneration and diabetic retinopathy. I work on Stargardt disease, which is one of the most commonest macular dystrophy. So I just wanted to, you know, update that we have already designed our dual vectors. We have finished our animal-based experiments. As Dr. Pooja and Rohit sir team is soon going to start the corneal-based gene therapy for LOX gene. It will soon be followed by a gene therapy human trial for the ABCA4 gene. So that is I wanted to comment. If any of you are interested in collaborating with us, if you have a patient for there is no treatment, syndromic features, degenerative diseases, please get in touch with any of us. So we have a team who are here to, you know, help guide this patient, track the patient, collect the samples, and put it into this chain of research so that we can offer whatever the best we can. That's my.
Yeah, thank you very much. So about, you know, samples, it depends on what study you want to do. So if you are doing a genomic study, you have to, you know, store the DNA, and that protocol is different. And you want to store the tier for proteins, then that storage methodology is very different. So therefore, you talk to a scientist, a basic scientist, before you start to store your patient samples. Each one of them have a different protocol. Okay, it's not that you can store the RNA the way you store the protein or the way you store the DNA. Okay. Yes.
Madam, about the binarization process you said about the macrophages, is it a software? Madam, or is it available as a software? Because there are many diseases now, glaucoma, venous occlusion, why these macrophages are now under the scanner.
Yeah,
So
so actually,
how do you get that binarization? Is it available for common use?
no, it's not available as a software, but we use the ImageJ software for analysis. So the images are procured using the OCT machine, the spectral domain OCT, and but we are manually segmenting those images, and those images are then we are transporting them to ImageJ, and then we are using all this segmentation protocol and the binarization. But it's not available as a software, but it is described in the papers. There is one by some Ong Ji et al. They have described, and we are also our paper is also under review. So we have also described the method in detail. But
in that study, they had actually done a 3x3 scan, and they had avoided macular edema.
Yes.
But
That's because it's difficult to segment in a macular edema because you have a thickened macular edema. But you can manually you can do it. So that's how we have done it manually.
Thank you, ma'am. Thank you, Dr. Edwin. We move on to the next speaker.
So Dr. Sundaresan will be the next speaker, and he's going to talk on mitochondria and wound healing is the topic. And we are running behind schedule quite a lot, and therefore requesting Dr. Sundaresan and Dr. Tandon to give a short talk, so that by 4:45 we'll wind up and leave this hall for the next session. Thank you.
Okay, thank you, Dr. Kumar. So I will, lack of time, I briefly emphasize my point. So I'm going to emphasize 3 points. One is what we did for the past 25 years related to corneal genetics. Second one is what we did for the mitochondrial research for the past 25 years with the various eye diseases, and what is the interaction between the mitochondria and wound healing. So concern with the cornea, you know the endothelial dystrophies, there are 3 types, CHED, Fuchs, and then PPCD. So in 2006, till 2006, the candidate gene is not identified. Our research group, in collaboration with the Singapore Eye Research Institute, we identified the candidate gene for CHED2, that is the autosomal recessive disease. The candidate gene SLC411 is present in the 20th chromosome. So we did all the expression studies, and then we cloned the gene, and then we expressed the gene, and then we did the cell culture model, Western blot, and everything. We proved, yes, this is the candidate gene for CHED2. This data was published in Nature Genetics. I'm one of the first author in this paper. Second one, concerned with the Fuchs endothelial dystrophy. There are a lot of candidate genes are there, but there is again SLC411 is a candidate gene. There is a crosstalk, but the collagen gene, collagen 8 is very, very important gene for this particular disease. So we entirely screened, and then now we have a spectrum of genetic variation for Fuchs endothelial dystrophy. And then macular corneal dystrophy. This is very recently our publication from Molecular Vision. We screened more than 130 macular corneal dystrophy patients. And then concerned within the Indian population, there are 3 types of macular corneal dystrophy. I mean, the types, phenotypes. One is the type 1, 1A, and then 2. So in concern with the Indian population, the type 1 is very, very common. So that's what we reported through immunohistochemistry. And then concern with the mitochondria, because I'm going to correlate with what is the role of mitochondria in cornea. So concern with the cornea, we extensively studied what is the interaction between the mitochondria and the nuclear genes. So you have to understand the nuclear gene is separate and the mitochondrial gene is separate. In the mitochondrial genome, there are 13 genes are there. And then the 13 genes is interacting with the nuclear genome. We have 19,000 genes, and then depending upon the disease, there is a crosstalk. Concerned with the first paper, primary open-angle glaucoma. There are primary open-angle glaucoma, and we studied what exactly the mitochondrial crosstalk with the mitochondria and primary open-angle glaucoma. We published in a very high impact journal. In addition, the right-hand side, so we taken the cadaver eyes, and then we segregated control diabetic and diabetic retinopathy cadavers. From that, we take the neural retina, and then we studied what is the exact role of mitochondria in the mitochondrial pathogenesis. That also is the entire more than 32 pages publication is published in Mitochondrion. And then concerned with the mitochondria for the past 15 years, concerned with the Leber hereditary optic neuropathy. So what exactly the primary mutation, what exactly the secondary mutation, what is the crosstalk between the nuclear genome and mitochondrial genome, and hospital-based studies. Because whenever we refer, we are quoting only the Western population. Concerned with the LHO and Leber hereditary optic neuropathy, 95% of the patient positive for primary mutation. In our case, is different. So that's what we are looking the entire what is the crosstalk between the mitochondrial genome and nuclear genome, where exactly the nuclear genome is interacted, whether in the complex one or in the mitochondrial dynamics. We extensively studied for the past 15 years. All these papers are published. Please, go through our publications. So, concerned with the cornea, concerned with this particular topic, wound healing. So, recently we made a study design, and then we are preparing a study protocol or study design. That time, I came across this very good paper. It is published in AIOS very recently in 2024 from Israel. So here, I think I don't want to go line by line. The content is you take the corneal cells, the human corneal cells, and then you isolate the mitochondria from ARP cell line, that is the retinal pigment epithelium cell line. From that, you can isolate the mitochondria and then incubate that mitochondria with the corneal cells. When it's incubated, it means the exogenous mitochondria, it is entered into the cell, and then once it is entered into the cell, it increases the ATP production as well as the membrane permeability. So due to this, the wound healing process is very high. So that is the point. So here, in addition, they made an animal model in the sense they made an alkali burn in mouse model, just exactly alkali model, and then in the mouse model, and then they isolated the mitochondria from the liver of the mouse, and then now they have the mitochondria of the mouse from the liver, and then they added topically, and then they are checking how it is the wound healing, and then the the corneal thickness, they are measuring that one. You can see here in the figure 1, the green one is nothing but a kind of mitochondria is inside the RPE cells. From that, we can able to isolate, and then you incubate with the epithelial cells. You can see the green one, the exogenous mitochondria is entering into the cells, and then the green signal, you can see it is entered into the mitochondria. The control, it is not entering. So that is the flash slide it is representing here. In addition, this is the wound healing, the control, and then mitochondria. This is nothing but a kind of corneal cells. You can plate it in the 0 hour, 24 hours. In the control group, there is no proper wound healing. In the scratch model, the mitochondria is in the sense you have to apply the mitochondria, whatever you isolated from the liver cells, and then you can see the wound healing closure percentage. You can see the second histogram. The mitochondria, the percentage is very high. It means the wound healing process is very high when you incubate with the mitochondria. And then concerned with the proliferation. The proliferation of the cells also is increasing when it's incubated with the mitochondria. You can see the difference between the control and the mitochondria. Obviously, the proliferation is very good, meaning it is enhancing the wound healing. Then this is again, it is in the animal model, how the mitochondria is entered into the cornea. That is the kind of representation. They have put a specific marker to detect the mitochondria. You can see it in the yellow color, and the green one is the nucleus. Here, as I told you in the animal model, just alkali burn, you make alkali burn, and then you add the mitochondria topically, and then you can observe day 1, 2, 3, 4. In the first panel, the control, there is no clarity, and then you can, within the 4 days, the corneal is recovery, promotes the corneal recovery. And then the wound healing, the graph, you can see the same thing representation within the day 4. When you add mitochondria, I think the wound healing process is perfect. So, and then they are measuring the corneal thickness. In the control, the corneal thickness is not that much reduced. When you add the mitochondria, the corneal thickness is reduced. So based on this, the wound healing, when you add mitochondria, is playing a major role in the sense it is once it is entered into the system, the cell, it is increasing the ATP production as well as the cell permeability. Based on that, the wound healing process is perfect. So I think this is the conclusion. I don't read the same thing. I already emphasized. And then the conclusion is the mitochondria promotes the corneal re-epithelialization and wound healing. The augmentation of the mitochondrial levels via mitochondrial transplantation may serve as an effective treatment for including the repair of corneal epithelial defects. So this is the points I would like to emphasize. concerned with the gene therapy for cornea. So now, as I mentioned in the very beginning, so right now people are adopting the gene editing technology, either the CRISPR-Cas and like that. So there are so many techniques are there, RNA therapies, stem cell therapies like that. In Australia right now, recently I think there is a paper published in JAMA. Just they're adopting the RNA therapies for Fuchs endothelial dystrophies. If during the when you treat with the RNA therapies, definitely the progress is very good when compared to the control. That's what they're emphasizing. That kind of research is going on. So with this, I'll stop. Thank you very much for your attention.
Thank you, Professor Sundaresan. We'll have Dr. Tandon next, and she's going to talk on the, sorry, 1 minute, bioengineering the cornea.
Good evening, everybody. Thank you for inviting me to participate in this session. So I'm going to give you a little outlook about what we have done for bioengineering solutions. Not able to get it to move.
Excuse me, the slides?
Yes, yeah. Okay, so biomedical engineering applications with respect to cornea, let's put it at 3 levels. One is the expansion model, then there's 3D printing, and then there's gene therapy. Along with that, we also have applications of AI for diagnosis and analysis. For the purpose of this session, I'm going to focus on tissue engineering, and along with that, I'll say that when you talk about assisting recovery, we have also amniotic membrane. Let's not forget the amniotic membrane and its role in ocular surface reconstruction. So where did we start? This journey has gone on for about 20 years, where we learned that we can do a maximum manipulation of the cells outside the body and actually apply it on the human cornea and get a good result. But we ran into a very major hurdle when we came into asking for this to be accepted as a standard therapy. Since it was so complicated and it was not accepted as a standard therapy, then one resorted to SLET, which is autologous cells taken from one eye and just put on the surface inherently. And one should not disregard the learnings we had from the previous cultured epithelial protocols, because it gave us an idea as to what worked and what didn't work and so on. So effectively, it was the lab in the eye itself. The tissue culture lab in the eye itself is where we went with the SLET. So this is the CLED journey from bench to bedside, and we've all gone through these various stages, but it also indicated to us that whatever we are talking about, cell therapy, gene therapy, everything, it's ultimately going to require regulatory approval, and we have to push for the policy change on that grounds. What we consider as accepted in peer-reviewed literature, it may not always be directly translatable at the level of policy, because they want things at the industrial level. And unfortunately, cell therapy, gene therapy, etc., to get at industry level applications, you require a big company which is going to do it. And largely, it is the research labs and clinical research kind of a scenario where this is being applied. So it is going to be the big hurdle, because it is a, we can say, orphan disease kind of a level. It is a niche area, and no big company is going to be able to take it up, because it is just simply not financially viable. So we have to have a way out. So just to look over the various interventions, have we been doing things wrong by doing these protocols? Not at all. As you can see, if you look at the various outcomes of surgical interventions for the various types of therapies, whether it was SLET, CLET, corneal keratolimbal allografts, and so on, all of them had similar anatomical success rates and good functional success rates. So if you do a systematic review, you find out that everything works. It depends upon that surgeon and the patient and that particular hospital, what is available to you and what you would able to prescribe for that particular case. So with this experience we had, and whatever we set up with the help of government support under projects, we thought we take it forward to now looking at, can we proceed with bioengineering for reconstruction of the cornea. We do know that corneal blindness is very extensive, and we do know that there is a shortage of donor corneas. We also know that with allografts, there is a risk of corneal rejection, and not all of them succeed. So therefore, lamellar keratoplasties that we are using not suitable for surgery corneas, those corneas also, a large number of them in the country are not being used. So those corneas, if properly processed, can be used for other indications. So looking at the where can we go with the synthetic substrates, etc. We've also looked at substitutes for amniotic membrane, because there are challenges with amniotic membrane. It's not sterile, it's not, it has a shelf life, and it has a very stringent storage protocol. So can we use a scaffold or any other membrane in place? So we have looked at the use of polycaprolactone as a biosynthetic substitute for amniotic membrane. We have found that it is excellent biocompatibility. It supports the growth of epithelial cells, and this was important because if we put this amniotic membrane on the eye for an ocular surface burn, is it going to allow the re-epithelialization, or is it going to cause inflammation, and so on? So in the lab we have shown, so this is one place where we went initially in the lab where we showed the biocompatibility, then we proceeded to animal experiments, and we were showing that there is a good re-epithelialization of a chemical injury induced in rabbit eyes, and equally effective as a natural amniotic membrane. But again, we have reached that point, and then now how are we going to make it available? That is another story altogether. So tissue engineering approaches for corneal reconstruction, there again we have electrospan scaffolds, as I mentioned. We can use this PCL scaffold, again reiterating what I said before. And then we are also able to show that with this kind of a protocol of using a decellularized cornea, there was the less immunogenic cornea. So therefore, less vascularization and less immune inflammation after transplantation. So this is where we went. First, we have used cross-linked decellularized corneas in lamellar procedures in the rabbit. I would acknowledge that I used the Department of Neurosurgery animal lab to do the surgeries. And as you can see in this picture here, very well, beautifully integrated tissue, which has got no inflammation and well tolerated, and even the histopathology postoperatively showed that it was successful. So now are we going further with human corneas? So here I'll go give you an example of a surgery where this is the next thing that we want to do. So it is human corneas which were not suitable for optical transplantation, and almost 30 to 40,000 such corneas every year are being discarded in eye banks in India. And now can we use such corneas? So first and foremost, all such eye banks, instead of discarding such corneas, they should immediately put them into glycerol and make them available. So here's an example of a case who had an exposed haptic of sterile fixated IOL, and then instead of using fresh cornea, this is a glycerine preserved cornea, and you can see how transparent it is. So the anterior lamella is as transparent as the commercially available vision graft. So instead, we would like to encourage eye banks not to discard any cornea which is seronegative just because it has a low endothelial cell count. Put it into glycerol, and somebody somewhere will find a use for it. And this is, so now the question is, in this particular case, it was a glycerol preserved cornea. The other thing is, can we use the decellularized cornea? Because the glycerol preserved cornea is very simple to preserve, but can be improve the technology by using a decellularized cornea. And that is the next, as I mentioned in the rabbit pocket surgery. So now we are looking at producing that same product for human implantation. This is what it is. So we have a non-suitable for surgery cornea. At the moment, we're using tissues which were culture positive, and then it is processed. This is how it looks, so opaque when originally. And then after decellularization and cross-linking, you can see that you have a transparent tissue. And remember, we're not transplanting the full thickness cornea. It is only the anterior lamella. This is the bioreactor which is used to process the cornea for decellularization, and it's a reasonably transparent tissue. I don't think anybody will hesitate to use this tissue for antilamellar keratoplasties, for patch grafts, or even if they have to use a patch graft to support glaucoma drainage device. So in a nutshell, I feel that we can go ahead and we can use the corneas, at least the ones which would be unnecessarily discarded. They can be processed in this way indigenously and made available for transplantation. Thank you very much.
Thank you, Professor Tandon. We have to vacate this hall, so we don't have time for questions. So with that, we conclude the session.
Dr. Pooja, please come up. Dr. Trimalesh, Dr. Purna, Dr. Swami. One picture, please.
Yeah, so we'll have all the speakers, we'll have a group photo. And with this, we conclude the innovations in cell-based ophthalmology. I thank the speakers and, of course, the audiovisual and the panelists. And, of course, lastly, the audience. Thank you.